Morning,
Hello.
Alexander. How you doing this morning?
Okay. Good evening from Indonesia. So we have 12 different hours.
So let's see. So that will make it what? Early evening, right? Your time?
Yes, evening.
Good.
So
Well,
here is goes to what is it 7? I mean almost to the 8 p.m.
okay. Yeah,
here.
it's
Yeah.
I'm in Wisconsin, so it's almost 7 a.m. So
Yes.
I've got an alarm going off. I'll be right back.
Okay. Thank you.
I have alarms set all over the house so that I don't forget. Because it's a little bit unusual time for me too. So.
Okay.
So have you been able to sit in on the talk so far?
Sorry.
Have you been able to
Could
listen
you?
to many of the talks so far at the meeting?
Not really. I mean, I'm sorry if my English is not good, really good here.
That's all right. Yeah, I've had other commitments, so I've listened to a few, but not all of them. So
Okay, got it. And also, I'm sorry that I cannot attend. I could not attend yesterday because I have a sick for a few days ago, so I have to bed the rest. And now I'm good now, so I'm ready for present my result
good.
of graduate thesis. So I think we waiting for another participants, right?
Right.
So it's
So
only me.
so in about at 8 o'clock, there'll be some welcoming announcements. And then your talk is at 8:20. So do you want to practice sharing your screen? Just to be sure that that all works?
Oh, sure. Wait, I'm looking at this PowerPoint for first time first. Okay. Let me try to host disable participant screen sharing.
Right. So down at the bottom, there's a little button that says share screen.
Yeah, I know, but this it is say that host disabled participant, so
Oh,
I
okay.
cannot.
All right.
To
Let me
show my PowerPoint.
okay, so let me okay, see if you can do it now. I've made you a co-host.
Okay, I tried. My presentation is already show here. Is it also
Yes.
show to you?
Yes. Yep.
Oh, okay,
So
got it.
that's good. Okay. So
Thank you.
you can stop sharing now if you wish.
Okay.
Perfect.
Thank
Yeah,
you.
so I'm still learning at my end how to allow people to do different things. So that's why I'm glad you came on early so I could practice. what I'm supposed to do. So that's good.
May I know who are you? I mean, where are you from?
So I'm in the US in Wisconsin and have worked with a variety of water birds, but currently I'm working with an aquatic passerine, a small swallow that feeds
Oh.
on aquatic insects. So I'm a little bit out of the mainstream for the types of birds. So what birds do you work with? Or sounds like maybe a large variety.
Yeah, the typical bird that I work is heron, like a great heron, great egret, little egret, and lesser adjutant stork. The vulnerable one. Yeah. So this species is really hard to find. And thankful God I can find this species.
Good. Yeah. We have similar herons and egrets, but in the US, we only have one kind of stork, the wood stork, and it's just over in Florida, so not very many. I don't get to see storks very often, so.
So I think maybe like climate change or anything like what happened right now, such as the recent of wildfires, maybe can make the hard to find the wood stock maybe in the USA. Is it?
Yeah, there was an interesting paper yesterday, and partly because of climate change, I think they're moving their range farther north, and in more places now. So that's a good thing for them to be more spread out, because there's so many problems that these birds can have to face and overcome.
Interesting.
Yeah, it's very interesting. So are you having a lot of issues with sea level rise and some of your colonies, on islands and places?
About my work. So actually this work is I produced from my graduate thesis research in 2018 until 2019, and then there was oil spill in the Balikpapan Bay. So I will try to look at, is there any factor, I mean like the sea levels maybe, or the water depth and the patch area, and also the oil spill that influence the foraging habitat selection by these water birds in Balikpapan Bay. And yeah, from this result, I think this is a good how to say that, like a good recommendation to the government, because in Indonesia for the next time there are a new capital city in Borneo, and the Balikpapan Bay is not really far away from the new capital city, so it could be another next threat for the. I hope so. There is no very hard threat. I mean, like really, how to say that, facing to the species because of the next capital city. I mean, capital the capital city of Indonesia. I think you also heard, right? Or maybe just not really finding this news in the USA.
Yeah, that I had not heard, but you know, a lot of times we don't seek out and get news from as many other parts of the world as we probably should.
Mm,
But so that's good to know.
Hi, Chris. I think I'm actually earlier than I needed to be, but I just wanted to check my audio and video and slide sharing and stuff.
Correct. And I made you a co-host, so you should be able to share your screen. So if you'd like to practice that, and because I can hear you and presumably you can hear me and Alexander. So
Hi, Alexander. You live in, you're from Borneo, you said?
yeah, I'm in Surabaya right now, not in Borneo, but my field research was in Borneo.
Oh, cool. I was able to visit a few years ago for a conference. So beautiful. Also,
Yeah.
in some areas sad because of the palm plantations. Okay, let me just okay, here's my screen. So I will bring up my talk. Share my screen. Is that looking okay?
yes, okay. We've
Yes.
got do mirrors exhibit optimal foraging.
Yes.
Okay.
Yeah, great.
All right, perfect. Looks good.
Thank you. And there's nothing else I really need to know, right? Questions will all come at the end. All that good stuff.
Yes, and the way we're going to do it, Marissa is going to introduce everybody, and then I'm going to somewhat keep track of time. I'll try and if you haven't wrapped up by 18 minutes, I'll pop on and let you know you've got 2 minutes left, and then if there's time for questions, Marissa will facilitate those. Otherwise, we can wait till the end, and then if there are questions in the chat that we haven't gotten to, we'll encourage the people to just reach out directly.
Okay.
And email the questions. So.
And I am teaching a class at 10 a.m. So I'm not sure I'll be able to stay until the full end of the session. So I'll just put I'll make sure my email's on my first slide, but I'll add it again to my last slide too. But,
right. Yeah.
Right.
And if you put it in the chat, people can then come back and look at the chat.
Okay.
So that might be a good way, because people are coming on and coming off pretty regularly. So.
Okay. Thank you so much.
Perfect.
Can I turn off the video?
Yes, you can.
I have to do something.
I may turn mine off here in a second as well, because I don't have great internet connection at home. So.
Thank you.
Good morning, Gaurav. You're talking later this morning. Would you like to practice sharing your screen to be sure everything is working properly both directions?
Hello.
Yes, I can hear you. Would you like to share your screen?
Yeah, I will.
That way we'll be sure we're ready to go a little bit later this morning when your talk is scheduled.
Yeah. So, are you able to see my?
Yes, moving with the
presentation?
monsoon, correct. Very
Yeah.
nice. All right. Good. I think we can all hear you and see your screen, so you can stop sharing when convenient.
Yeah.
Perfect. Thank you.
Welcome.
Hi, Yvonne. I see that you're on. Would you like to practice sharing your screen to be sure that we're technologically got everything, all our ducks in a row, so to speak?
Hi, Michelle. Yeah, thank you. I just saw that you added me as a co-host, so that should work.
And that is a man who has an internet sexual harassment and making threatening gestures showed up. This man crossed state lines, in line at a two in Brooklyn store. You're up close and personal.
That looks good.
This isn't a message about hating black because it's never. Girl, they swear you hate black people. Girl, and
Does that work?
Yes, it does.
Okay, great.
Message about how we as a community love
Okay, I'm going to have to mute everybody. We've got somebody talking in the background.
We identify areas of toxic black masculinity that continue to threaten our lives instead of just allowing.
Welcome everyone to the Conservation Foraging and Movement session this morning on day 3 of the Waterbird Society meeting. We'll get started at 8:20.
And this is Chris. I'll be the official timekeeper, so I'll watch the time. And if you haven't started to wrap up your talk after about 18 minutes, I'll pop in and let you know you have 2 minutes left, so you can start to wrap up. As we've been doing throughout this meeting, if you'll put your questions in the chat, then Marissa and I will be watching that. And if there's time for questions, we'll pose those. And then if not, we'll please reach out to each of the individual presenters and ask the questions offline if we're not able to get them during the course of this meeting. Hi, Kate. I see that you've joined the meeting. We've still got a couple minutes before we start. So if you'd like to share your screen, I've made you a co-host, then we can be sure that we've got connections both directions.
Hey, Chris. Yeah, just give me a second. This is like my first virtual, so hopefully it won't be too bad.
Yeah, they're a challenge, but we're getting a little bit better at them as we keep having these sorts of meetings.
Indeed. Okay, so let's see. If I can.
So if you go to the bottom, there'll be a green button saying to share screen.
Right. I just had my PowerPoint up and it decided it wanted to start on me. So
Oh.
yeah. Let's see. Let's see more. Share screen. There it is. There it is.
Okay, spatial temporal variation and Black Skimmer migration.
That would be it.
Okay, great. All right. So I think we're in good shape. You can stop sharing, and I think we're a minute or two out before we're ready to start. So we've got all speakers. So I think we're going to be in good shape for this session.
Wonderful. Well, the time now is 8:20, and our this is again the conservation and foraging and movement session. Our first talk of this session will be by Alexander on waterbird foraging habitat selection in Balikpapan Bay, water depth and patch area as important factors. Would you like to share your screen now?
Sure.
We see that you started screen sharing, says double click. There we go. And would you like to go in presenter slide.
Can I start it right now, or maybe
Can you
wait
make it full screen? If you want to click on the button on the bottom, that makes it full screen, so we can see one slide at a time. Right now, we see your entire Microsoft PowerPoint program.
I have already showing the.
Maybe your audio is a little. I think your audio is cutting out a little bit as well. Chris, did we lose Alexander?
I don't see his PowerPoint anymore, so
Okay, you might have had connectivity issues. Maybe we can give him a little bit to come back in.
yes.
Okay.
Yeah, I don't see him at all on the participant list now, so I think he completely went off, and hopefully he'll be able to come back on.
Yes. I guess just in preparation, Gaurav, you're ready to go with your screen, with your PowerPoint?
Yeah, I'm
Okay.
there.
Okay, thank you. Okay, looks like Alexander's joining us back.
Okay, I'm sorry for the technical problem in my internet connection. I don't know, maybe it's
That's all right.
unstable internet connection here. Hello?
Yes, perfect. We're glad you could join us again. Yes, whenever you're ready, you can begin sharing and begin your presentation.
Could you able to me for screen sharing?
Yes, I've
Because-
made you a co-host, so you should be able to share your screen now.
Okay, thank you. Is it my screen is already show to your monitor?
Yes. So if you go to the top under slideshow on your screen, you should be able to do it full screen. But if not, go ahead and just proceed as they're showing now.
Okay.
There we
Yeah.
go. That looks
Yeah.
good. Thanks.
Okay. Thank you very much for all audience here. I would like to share our results about the waterbird foraging habitat selection in Balikpapan Bay, water depth and catch area as important factor. And this is my first time to show this result to the international audience. So I hope for misunderstand for my mistake grammar. So this the introduction. The Balikpapan Bay as the one of the feeding ground for waterbird in Indonesia, and we found that 4 species from the little egret, great egret, purple heron, and lesser adjutant stork as the one of the vulnerable species in Indonesia. And we saw that there is a like a congeneric species between the great egret and purple heron that we found see in the one area, one place. So when the foraging simultaneously for one of these area in one place, there is a competition. But when the foraging in one place, either the great or the purple heron, so one of them will use the different resources. So that's why there is a competitive exclusion principle in this habitat that lead to the habitat selection during foraging. And then as far as we know that the factor of habitat selection consists of 2 factors, intrinsic and extrinsic. The intrinsic one is food, while the extrinsic is accessibility to the biological and physical component habitat. If we look at the food as the intrinsic factor, of course, the waterbird is foraging, and the energy acquired by the waterbird will give the successful fitness. So that's why we try to look at the perspective of the energy from prey in the intrinsic factor. Is it the influence to the foraging habitat selection? And for the extrinsic factor, there are a biological component and physical component. In biological component, we look at the prey density that impact to the food resources, and will be the biological indicator in ecological process in this foraging process. So it's also influenced the foraging. So we would like to know about the factor and this physical factor like the water depth and catch area. Some of papers say that each species has their how to say that the access able to the water depth, like the great egret can look at the can forage the area when the water depth is 10 centimeter, while the lesser adjutant foraging on the 35.56 centimeter. So we would like to find out the water depth and pitch area. The pitch area we find out because some paper called that the pitch area is relevant to the foraging behavior of waterbirds. And in addition, there was a oil spill in 2018. So from all this physical appearance, we would like to know, is there any how to say that, the rule of this physical component of habitat influence the foraging process. So the aims from our research is to measure the foraging habitat selection of waterbirds from the Ardeidae and Kikonidae in Balikpapan Bay. The second aim is to examine which factors influence the foraging habitat selection. And the third one is about predict the model of foraging habitat selection. In material and method, so this is the maps about the Balikpapan Bay, and this map show that there are some wetlands that we have found, like a shrimp pond and the mudflat. So from the all 36 sampling location, as we found before, the 4 species we looked before in the introduction, we were sampling from the 13 December 2018 to 27 January 2019. We used the GPS receiver and compose all the data by using the QGIS. In the data collection, we have a few steps from the collecting the data. The first one is the 1-0 sampling. We use the ethogram from the many reference about the foraging in waterbirds. We use the one for successful foraging and foraging activity, while the score of 0 we use for no foraging activity and unsuccessful foraging. And the second step is we measuring the biotic data by using the coral sampling with a transect 200 times 20. And this coral sampling, we could collect the prey like a fish and crustacean, and we try to calculate the prey density, and also we try to calculate the dry mass and measure the S-free dry mass in the laboratorium in the my university from March until August in 2019. And the last step is measuring the abiotic data. We use the water market stick wood in centimeter measurement to measure the water depth, patch area by digitation based on the tracking data and base map we got. And the oil spill, we only try to look at there is a presence of oil spill or the absence. If the present, we give the score 1, while the absent we give the score 0. And there is the data analysis. We use the mainly standard selection ratio to look at the selection ratio from the foraging habitat selection by these waterbirds. And then we try to look at the normality test to make sure there is a normality between the selection ratio. If there is a normality, we try to look at the chi-square goodness of fit test to make sure that there is there any selection ratio that fit, or maybe there's no selection. I mean the preference. I'm sorry, the preference or no preference from the waterbirds. And we try to look at the confidence interval by 95. 95%. The second is about the factor. We use the generalized linear modeling framework. So from this all step, we try to look at the which one factor that can be used to the for influence the foraging habitat selection. So from the first result is about the foraging habitat preference. There are no difference between the foraging success proportion and foraging activity proportion. This means that there is optimize the foraging process from the waterbird that makes increasing the probability of foraging success for all these water birds. And this most of the water birds show the no preference. However, there also show the preference for 20-30 individual land like the shrimp pond and the mudflat for the little egret on the second picture. And then no, there is a 56 individual from the little egret. So this is the little egret has much abundant for the individual and give us to the about the preference of no preference and no preference. And also the all specials give the preference, but the interestingly the purple heron only no preference for shrimp pond and the mudflat. So from all 4 water birds, only purple heron that give no preference for any type of wetland. For the egret garceta or little egret, the things that we observe in the field, there is a hike in the industrial activity in the 2 industrial zone. The industrial zone also located on the mudflats, so this little egret doesn't did not give no preference for foraging habitat. There's a passing trucks that make these species stop due to the stress, run and fly away. While there is a unique one is the Gersik mudflat, and this is Gersik mudflat has no industrial activity until now. So when we observe the Egreta gersita, there are no how to say that they keep foraging because no noise of passing truck. So that's why we observe and we measure that the species has a preference for the Gersik mudflat with the no industrial activity. And also for the number of individual, we observe that there are only shrimp ponds, not in the mudflat, because on the mudflat we have showed before for the shrimp pond, only fewer individuals for foraging makes this species has a less probability of intraspecific competition. Sometimes the individual become aggressive and chasing away and barking to the another individual. So that's why make the increasing the probability to successful foraging. This individual will protect the foraging habitat by visit and forage more continuously after aggression to the another individual. So the little egret show the preference of the shrimp pond as foraging habitat. And now we look at the great egret. There are preference only in the shrimp pond rather than the mudflat, because we observe that this great egret only forage the shrimp rather than the another like a fish or anything, because more shrimp in the shrimp pond. And the other paper support that great egret also choose ponds with the crayfish, like the relative species maybe like in the stream, because they are crustacean. While the population of the species become larger, the pond size also extending over 19 years. So this species prioritize the crustacean as first prey to avoid competition. This is the congeneric species between the the great egret is a congeneric species to the purple heron here one. The purple heron only use the prey as their primary food resources, while in the ponds there are only less fish abundance. So that's why the purple heron have no preference to foraging in the shrimp pond. And then the fish abundance, while the less than shrimp abundance makes the prey availability for purple heron is less. So that's why purple heron try to leave the shrimp pond, while the great egret continued to forage the shrimp in the shrimp pond. The other species like the lesser adjutant stork or Leptocylus javanicus, the species we observe, it's only found on the one place is Gresik mudflats, and we catch the moment that the species catching eel when mudflat was rising. So we assume that the species will look at the more abundant dying fish, and also caused by the local trap we call zero. And the plus for this place is there is no industrial activity, so that's why I make a preference of foraging habitat to Leptocylus javanicus or lesser adjutant stork. And the most significant proportion that we try to measure is fish that trapped by the local trap, or we call it zero. And here is the model. We try to look at the more variables, and then from the multicollinearity, we found that only 1, 2, 3, 4, 5, 6, only 7 variables from I think 11 or more variables that we collected. And then for the all variable, we try to building the model and try to select the best model, and we have 2 best model like M1 and M2, and we try to fitting the 2 selected model in the next step. And we've got there are 2 factors that consistent not consistent to give the how to say that positive response by look at the significant variable. So most of the variable in M1, like a presence of oil spill, fish density, and crustacean density, have no significant variable to the foraging habitat selection, while the patch area and water depth in M1 is consistent to give influence to foraging habitat selection, as well as in the M2 or model 2, while only patch area and water depth as the only predictor variable in the second model. Here we show the model foraging habitat selection, and this is the M2 that we choose because the M2 only show the more simple predictor variables rather than the M1, because in M1 there is 3 variable that no significant to the foraging habitat selection or to the response variable. So we choose the M2 as the best and fitted model, like here.
Alex,
So,
you've got just a minute and a half left or so on your time.
Okay, thank you very much for your reminding. So the patch area and water depth we use is the most important factor in this model, and some paper also support that the suitable habitat like a Gersik has a similar in suitable habitat for stock in China, like a high patch concentration, low fragmentation, and minimum impact. And this most of paper we find out there are also have evolved research in the connectivity and also in waterbird normalized difference water, that there are local effort by by mapping the habitat suitability model in the other city. And this result we provide the simple mathematical equation model. So from our result we recommend that the mudflat and shrimp pond in Gersik has to be waterbird sanctuary area, and the keeper can collaborate to the government to to keep the waterbird sanctuary area and protect it. And then the keeper can be guide for off-it tourism to make a growing economy for the local livelihood. And we hope there are subsequent field work in the whole Balikpapan Bay. So the potential habitat we try to address to the government. So we hope the government to regulate the policy to reach SDG, and also to make sure that there is no harm to the area due to the next plan for capital, the new capital city in Indonesia, changing the Jakarta in this area in the Balikpapan Bay. So this is the conclusion about our research. There are little egret prefer to forage in the both habitat. The great egret only show preference to shrimp pond. Purple heron decide no preference, while the lesser adjutant stork choose the mudflat. So we have patch area and water depth to influence the waterbed foraging habitat selection. And the last is the best and fits model is the selection ratio from the model as the variable response, while the large patch area and shallow water depth become variable predictor. So thank you for the chance to present in this forum discussion. Thank you.
Thank you very much, Alexander. We'll take questions for his talk at the end of the session to just stay on track. So next up is Gaurav, who will be talking to us about moving with the monsoon. Gaurav, whenever you're ready, you can share your screen and begin your talk.
Hello, is my this presentation visible?
Yes, it is. Thank you.
A very warm good evening to one and everyone. So today I am here to present my work on the flamingos from the Western India. So as we all know, the flamingos are one of the most ancient birds, and in total there in the whole world there are 6 flamingo species. And when we come to the old world, there are 2 flamingo species are there. One is the lesser flamingo and another one is the greater flamingo. So basically the lesser all the flamingos are water inhabitant birds which prefer to wade in the saline and freshwater bodies and along the coastal lagoons, mudflats, and the salt pans. When we talk about the 2 species, lesser and the greater flamingo, as the name suggests, the greater flamingo is one of the tallest flamingo species found, and whereas the lesser flamingo is the smallest flamingos found. According to the diet spectrum, the greater flamingos are generalists. They feed upon the wide range of these cyanobacteria, algae, and crustaceans, whereas lesser flamingo is a specialist which is extensively feeding on cyanobacteria. As per the IUCN, currently the greater flamingo is a least concerned species, whereas a lesser flamingo is a near threatened species. When we come to India, we have the two flamingo species present in India. When you talk about this greater flamingo, the greater flamingo is widely distributed across the whole India, and the area which is shown in the purple color are the areas where the resident population of the greater flamingos is found. When we come to the lesser flamingos are mostly concentrated towards the western part of the India. And again here in the Kutch district of Gujarat, state of India, we have a resident population of flamingos. Moving further, for this flamingo work, we selected the Kutch district as our study area. The main reason for this selection was that the Gujarat state is one of the stronghold for the flamingos, both the species of the flamingos in India, and the run of Kutch is the important breeding site for both the flamingos in the whole Northeast Asia. So in order to know how the flamingos are using this landscape, we selected this area for our study. And moving further, we had the two major objectives for this study. First was to understand the fine scale movement and ranging patterns of the flamingos during the breeding and the non-breeding season. Second, we wanted to know what are the habitat preferences of flamingos in the Western India. To meet with these objectives, we set some research questions, which were what are the ranging patterns of the flamingos, and in these ranging patterns, are there any seasonal variations in the ranging patterns, and what are the factors which are influencing these variations in the ranging pattern. And we also wanted to know what are the habitat, what are the critical flamingo habitats in the Western India. So to fulfill these objective, we initiated capturing and tagging of flamingos in the Kutch district. For capturing, we used two methods. We use noose traps and mist nets for capturing of the flamingos. And upon capture, the flamingos were fitted with Ornitrack 30 GPS GSM backpack transmitter, which weighs 30 grams, and the transmitter weight was less than the 3% of the body mass of the flamingos. And this transmitter was attached to the back of the flamingos using a Teflon harness. For analysis of home ranges, we used kernel density estimate, and it was performed in RStudio using a package called KS package. So in all our tagging effort, we were able to tag 4 lesser flamingos and 2 greater flamingos. The tagging activity was initiated in the month of May and June in 2019, and till since that time we have 4 flamingos which are still active and transmitting data, whereas the 2 lesser flamingos which we tagged, they gave data for only a short interval of time, and the tags was malfunctioned. When we move forward to see how the flamingos use this whole landscape during the tracking period, we interestingly find that the lesser flamingos, they were using these eastern parts of the Kutch districts extensively during their breeding and non-breeding periods. When we move to the greater flamingos, basically use the whole landscape in a much different way. They were moving very frequently from eastern to the western parts, and from southern to the northern parts. And one of our flamingos visited outside the country. It visited the Pakistan, a small coastal wetland there. And during the whole trekking period, none of the flamingos left this Kutch district where we initiated the tagging activity. When we analyze this data to see how the home ranges varied across the seasons, we found that during the pre-breeding season, the flamingos were the lesser flamingos was using these Gulf areas extensively, And in all these three, all the four flamingos, lesser flamingos tagged, they use these Gulf areas extensively. Whereas during the breeding season, we found that they uses the run habitat extensively, and during the whole breeding period, they are only there in the run habitat. They don't use any other water bodies at that present at that particular time. And post breeding season, they flamingos are they return to their pre-breeding areas and stayed in their location. So when we move towards the greater flamingos, we found that the greater flamingos, they also show the similar pattern during the pre-breeding and the post-breeding season. Whereas in the breeding season, they were spatially segregated from the breeding areas of the lesser flamingos. Like the lesser flamingos use that eastern parts of the greater run of Kutch, whereas the greater flamingos, they were limited to the towards the western part of the greater run of Kutch. We analyzed these home ranges and found that during the breeding season, the home ranges of all the flamingos significantly increased in comparison to the pre-breeding and the post-breeding season. And if we see, if we compare this breeding season of 2019 and the breeding season of 2020, we found that the flamingos, the home ranges expand a lot during this breeding season. Upon further investigation, we found that one of the factors which influence the habitat use of flamingos during breeding and non-breeding period is the rainfall. And if we see the trends of rainfall in Kutch districts from 2018 to 2020, we see that there is a significant increase of rainfall in 2020, so which influence the habitat use of the flamingos during the breeding season, because during the breeding season only these flamingos are into the run, and run is a quite dynamic habitat which get flooded by the rainfall and the rivers, which flood these areas only during the monsoon. And the extent of the rainfall during the monsoon thus regulates the time spent by these flamingos in to this habitat. So if we see in 2020 that there was more rain, so which resulted in spending of time in these habitat by flamingos, a period much prolonged time they spend into this particular habitat. If we further analyzed how and what were the wetland types which were used by the tagged flamingos, and we found that the greater flamingo, they extensively use the coastal wetlands and inland wetlands, whereas the lesser flamingo, they proportionally use all the three type of wetlands available in this landscape, which includes salt pans, inland wetlands, and coastal wetlands. Upon further investigation, we found that the four flamingos, the two greater flamingos, they were quite different in their habitat use. The one of the greater flamingo, it extensively used the coastal wetlands, whereas another greater flamingo, it extensively used this inland wetland during the whole period of the non-breeding season, which indicates that the two flamingos species which we were able to capture and tag, they belong to a certain different populations of the greater flamingos itself, which are specialized in their habitat. Whereas the lesser flamingo, if we see, they were more or less the same. All the habitats were used by the two flamingos in an equal proportion. Whereas, and interestingly, we found that the salt pans are quite essential for these lesser flamingos in this landscape, and the lesser flamingos are extensively using these salt pans. Moving further, we investigated how the movement was influenced by the rainfall. So what we did is we find out that net displacement of the flamingos from that initial tagging site. So we found that like as the rainfall is influencing this displacement from their non-breeding ground to the breeding ground. So here if we see that when the rainfall here if you see that when the rainfall is increasing, that movement amongst the flamingos is also increasing. At this particular time, the flamingos are moving from a non-breeding ground to the breeding ground. And again, if we see, we have two peaks in this displacement, and these two peaks comes during only the breeding season. So we conclude that from this information that during before as the monsoon is onsetting, the flamingos are moving from their non-breeding grounds to the breeding ground, and this extent of movement is being heavily influenced by the amount of rainfall, because the amount of rainfall ensure that the breeding grounds are available for flamingos to breed. And these run habitats are quite dynamic, as I earlier told. This dynamic habitat, they remain wet only when the monsoon is there. When there is no in the years when there is no rainfall, these habitat remain totally dry, and they are not available for flamingos to breed. So at that particular time, the flamingos are not visiting these areas. Further, from the tracking activity, we conclude that the flamingos which we tagged were primarily using the Kutch landscape, and they are likely the resident breeding populations of the flamingos. However, this region is quite important breeding ground, and sees a large congregation of these birds during the breeding season. And further tagging and tracking activity is required to be done in this area to target the migrant population, so that the use of these habitats during the breeding and non-breeding season can be much better understood. The Kutch landscape is a very important part of the flamingo ranging area in the whole Western India. And these areas were has been identified as a quite bird important areas. And in these areas, the movement patterns is also influenced by the flooding of the run habitat, and this run habitat flooding is in turn influenced by the amount of rainfall the area received during the monsoon season. And we found that during the breeding season, the two species are using the run habitat, but the habitat in this run habitat, it's spatially segregated. The lesser flamingos are situated into the eastern parts, whereas the greater flamingos are using the western parts, basically the area which is popularly known as Flamingo City, and is one of the most largest flamingo breeding ground in the whole Asia. And during the non-monsoon, non-breeding season, the birds tend to use the wide variety of wetlands, which include inland wetland, coastal wetlands, and the salt pans. I would like to thank PowerGrid for funding this study, and I would like to extend my thanks to Waterbird Society for providing me an opportunity for presenting my work into this conference. Thank you.
Thank you so much, Gaurav. We have time for a short question. If not, then we can take them in the end, or you can type them in the chat. So we can move on to our next talk by Yvonne. We'll be presenting on recent advances in the ecological study of the endangered Diablotin Black-capped Petrel at sea distribution, diet, and threat exposure. Yvonne, whenever you are ready, you can share your screen and begin your talk. Thank you.
Okay, thank you. All right, thank you for having me. Oh, wait, it's not sharing. Okay, it's full screen now, I'm guessing?
Yes.
All right, great. All right, thank you for having me. So I'm going to talk while we're going out at sea, moving out from the shores and flamingos to black-capped petrel and the pelagic waters of the Atlantic. I want to acknowledge my collaborators, Patrick Jodes of the South Carolina Crop Research Unit, Brad Kidd of American Birds Conservancy, Chris Gaskin of Northern New Zealand Seabird Trust, Gemma Lucas of Cornell, and Sarah Jensen of USGS. So the black-capped petrel is as I just said, a pelagic seabird that breeds in the Caribbean and uses the Western North Atlantic. Its IUCN status is endangered, with the population side that's decreasing, and estimated between 1,000 and 2,000 pairs and about 5,000 individuals. We know of 3 subpopulation. We expect maybe 10, depending on where they could breed. And we know only of 110 nests that have been found so far. Of importance for this talk is the fact that there are several phenotypes, or there is a range of phenotype within the species. 2 color forms, the light and the dark, so I'm going to mention that a lot, with intermediate. So this is a continuous range. It's not one type or the other. The light form is heavier in average, average 416 birds that were collected, and the dark form is smaller, lighter and smaller as well. There are genetic differences. Many show that the light and intermediate forms are closer together than they are to the dark form. And based on that presence in Western North Atlantic and on moult patterns, we suspect that there is either seasonal or spatial segregation during breeding. So I'm going to present recent research on the black-capped petrel. Distribution based on at-sea tracking, and then 2 smart study on diet and mercury. Starting with distribution, funding was from the Co-op Unit, ABC, and the Mohammed bin Zayed Species Conservation Fund. So what we know of distribution of black-capped petrels is from mostly ship-based surveys. Those were in the 80s and 90s, and then starting in the 2010s with the AMAPS effort, mostly in the US EEZ along the southeastern coast. And those data sets show 2 hotspots, one around Cape Hatteras. I don't know if you can see my little arrow, but to the middle of the US coast, and one further south near what's called the Charleston Bump. And those data were used to predict distribution in the US EEZ. And then more recently with the GOMAPS effort in the Gulf of Mexico, that confirmed the use in the Gulf of Mexico by black-capped petrel. So there's a new study by Jolis et al. That just came out on this. We also did individual-based tracking. So just to show you the differences in data sets, in April 2014 and 18, we tracked only 6 birds, 3 and 3. Those are the yellow and orange dots up to the right. And what this showed is that there are actually 2 different areas that are being used for chick rearing, because when we track them, that would be in the Southern Caribbean Sea, where there's an upwelling, and then during non-breeding season, using the Western North Atlantic and the Gulf Stream area. But there are limitations to those data sets. Very briefly, and main one for these talks, at-sea survey do not record phenotype, so we cannot use that. We cannot look at phenotype. And then tracking so far is only of the dark form and also very limited sample size, and only from breeding areas. So we were trying to look at a different angle of this question, and could we track petrel from marine location and locate unknown nesting areas? And would these nesting areas, would there be phenotype specific? Would the light morph breed at a different location than the dark morph? And the hypothesis being that both forms would have similar non-breeding distribution, but distinct breeding distributions. So to do that, we head out at sea in May 2019. And thanks with Captain Brent Patterson and Kate Sutherland, we used the Stormy Petrel, which is an offshore bird watching, pelagic watching boat. And they know the area very well, and they brought us to hotspots of black-capped petrel off Cape Hatteras. We use an inflatable and chum, so which is just a ground-up fish to attract petrels, and then a net launcher, which is usually used to tag whales, but was adapted by Chris Gaston and his colleagues to launch a net, and that uses compressed air from a dive tank. Once the bird were caught, we tagged them with solar PTTs, about 7.5 grams. We use sutures that on the back, that because we wanted that to last only a limited amount of time. The average of the tags were about 2.2% of body mass, with 8 geotrack and then 2 microwave tags were donated to the study. And processing time about 80 minutes in average. So in terms of results, we caught 10 petrels, 3 females and 7 males, that we didn't know ahead of time. And that we targeted, we tried to catch light forms in particular. So we had 5 dark forms, 1 female, 4 males, and then 4 light and 1 intermediate, 2 females, 3 males. And those were grouped into a single group for analysis. There were differences that were significant for wing chord and common length, the light form being bigger, and also those were molting 2 or 3 flight feathers, whether primaries or secondary at a time in May. I want to point out that tracking duration there is a range of tracking duration that is common with attachment method. So between 11 and 255 days. So 3 birds were tracked for only about 2 weeks. And so, yeah, this study is small. It's just 10 birds and then only 7 birds which were tracked for a good amount of time. So from Argus data, I used a random walk fitting using the package Foraging R, and I retained about 4,000 locations with a precision of 5 kilometers. They'll be useful later in the analysis. So all of the birds used the Western Atlantic. 1 bird wandered off to about the mid-Atlantic range. Unfortunately, the tag stopped working at this time. And 2 birds went south to the Caribbean. I'll come back to that later. Comparing to previous data sets, so it's consistent with previous data sets. In blue is the AMAP survey, the yellow is the tracking data. But it's different in 2 ways. Our data set was more northerly and pelagic. They did use the Gulf Stream around from Cape Hatteras to southeastern US, but they also used much more pelagic and eastern areas. And there is a limited use of the Charleston bump, which is the area in blue in the map. And the more northerly and pelagic distribution is mostly due to the light form, the light phenotype. So phenotypes have distinct distribution, significantly distinct, with the light form being more northerly and easterly, and the dark form being further south and west along the Gulf Stream. Generalized linear mixed model show that there is no effect on date, so it's not the fact that it's linked on duration time, and a small effect of sex and just on longitude with female, but that was only 3 birds being more westerly. In terms of conservation, so the US hosts most of the use areas for either forms, 100% of the core area for the dark and 3/4 of the core area of the light form. And then between 60 and 80% of the home range for both forms. So that's very important for conservation in the US. Moving on to nesting areas. So using location, just the Argus location, battery levels, and communication schedule, we were able to tell if a bird was or not using a burrow in the Caribbean. So as I said, 2 birds went south. To the right is the light morph in yellow. It went south around the beginning of October. And we suspect it used the location marked in F in the central map, which is Valle Nuevo in Dominican Republic. Based on low battery levels and lack of communication schedule for 2 weeks, we think it went in under the canopy and cleaned its burrow there, and then went back to sea for pre-breeding exodus. And then a dark morph came back 1.5 months later to a known nesting area in Haiti, a known nesting area of the dark morph. So there is allochron in Saint-Patrick. So there is a temporal but not a spatial breeding segregation. That's one of the main results of this study. Moving on to species vulnerability. So we, as a first step to studying vulnerability, I looked at exposure of two marine threats. That's spatial overlap at the macro scale. Also small sample size, but on the core area, there's marine traffic was more important in the dark than the light form. Marine traffic is linked to attract petrels to lighted vessels, and also pollution. Mercury and plastics were also higher in the dark form. And fisheries were higher in the light form, and that's based to one area around George's Bank that has a lot of trolling, fishing trolling there. And then finally, there was overlap with marine energy. So oil and gas in Canada, I'd say there was overlap with the home range and observed because we actually had locations within a lease area in Canada. And for wind energy in the US, it's modeled because it was only the kernel density that was overlapping the North Carolina lease area in the US. Moving on to diet, I see the clock spinning, so I have to get going. We looked at, did a DNA metabarcoding of fecal samples with Jenna Lucas of Cornell, funding from Birds Caribbean. So the only study of diet in the petrel is based on Moser-Lee in '92. They looked at crops of 57 birds, and squid was present in 93% of samples. But squid do accumulate in digestive system, so we want to use a different method to look at that. So we collected fecals opportunistically at breeding sites and at sea, extract DNA, and then Gemma did a high throughput sequencing. And using universal primer, we actually saw a limited occurrence of cephalopoda, so only in 2 samples out of 6, very small sample size again. Fish was present in all samples, and Magnidaria, which we think is a secondary feeding, eaten by a prey of a prey, in 1 sample. Looking at fish primer, so there was a lot of diversity, 10 species identified, but 57% of all sequence reads were not identified. We saw a difference in diversity between breeding and non-breeding, so maybe the breeding might be due to a more coastal distribution, in the Caribbean Sea. And also based on fish type, so 3/4 of sequence reads were from deep sea species. About 1/4 was 3/4, sorry. 1/3. 1/4 was from fish that perform diurnal migrations that come up the water column at night and then go down during the day. And then 3 species were fish that were they're fished commercially, snapper, scat, and runner. But small sample size, so we need to expand the sample size so we can actually find clear patterns and hypotheses. Moving on to mercury with Sarah Jensen of USGS and funding from the Seabird group. So Black-capped Petrel, just like any other Pterodroma, is highly susceptible to mercury accumulation through diet mostly, cephalopods and deep water prey, and that has impact on fitness and development. So we use feathers that we collected during other efforts. They were washed and digested, and we looked at total mercury. There was only a single previous study that was never published, and that showed that petrel were still within the first higher tier of exposure within Pterodroma. And this is our study, so it showed a yet higher level of mercury, but also a bigger range of mercury exposure within the samples. So there was 20 samples. So we didn't observe effect of phenotype, although exposure was different. We didn't see this effect in actual feathers, but there is a minimal sample size for the light phenotype. So next step will be, well, to increase sample size. So also hopefully increase funding, but distribution on diet and mercury. We like to look at ecological niche because of the different habitats that suggest different ecological niches. And then try to track the birds throughout the annual cycle and at finer temporal scales. I'm just mentioning a different study by Kate Sutherland. She's looking at historical changes in mercury in the petrel. Thank you to Brian and Kate for leading the trip, taking us offshore, and then Ari and Armin gave tags, and then support from our funders. Thank you.
Thank you so much, Ivan. We might have time for just a short question. If not, then we can, again, just have them at the end and move right on to Natasha, who will be speaking to us on the individual variation in the foraging personality and plasticity of an Arctic seabird. Natasha, whenever you're ready. Thank you.
Morning, everyone. One sec.
What do you get?
I got lots of keys and my usual stuff on death. This is a slide for girls.
Sorry, I'm having my
Looks great. You are on mute though.
You'd think I'd be better at this by now, but still struggle sometimes. Okay, so again, my name is Tasha. I'm an assistant professor at Gettysburg College, which is a small teaching college in Pennsylvania. And I'm going to talk to you today about a project that I've been working on with colleagues at McGill and at Trinity. So first, I wanted to talk a little bit about the framework that this research is kind of being conducted in. So I'm really interested in interspecific variation and how that actually scales up to influence population level and even community level dynamics. And I think this is a really timely thing to be studying. A lot of you obviously agree. There have been a lot of talks on individual variation at this conference. But the idea here is that particularly for long-lived populations, evolution can't really keep pace with the current scale and rate of current environmental change. So if there's variation within a species, and particularly flexibility across environmental contexts, that might give that species some ability to adapt to environmental change. So to ask these kinds of questions, the type of data you need are individual characteristic data. So things like diet data from isotopes or from provisioning watches, morphometric data. So things like the adult size or the chick growth rates, and then movement data. And of course, there are a lot of different tools we can use. We've heard about a lot of them at this meeting. Then those individual characteristics also need to be considered in the context of the environment. So this could be oceanography, sea surface temperature, chlorophyll A data, fisheries data, or prey data, information on prey biomass. And then ultimately, where this becomes really interesting is how this then links into demography. So actually using information on the flexibility of individuals to help predict how that, once we know how that influences fitness, to help predict how the whole population will respond given that information. And of course, classic tools like nest check data or survey data or marker capture data can be used here. So this is kind of the general framework. We're not going to be talking about all of these things today, just a portion of these things. But this is the framework we're working in. So the case study we're going to be using is on thick-billed murres in Coates Island, Hudson Bay. So this is a field site in the Canadian Arctic. It is, this is the location of the island here. It's definitely a ice-driven system. So the ice starts to recede, and that's when the seabirds actually move in and to begin to breed. Just to give you an idea of the scale of this system, the island across is about 130 kilometers, and the birds never really forage any farther out than about 100 kilometers from this site. The thick-billed murres, they are serial monogamous that have one egg at a time, and they can live to be up to about 30 years old. They generally move in around late May and lay eggs in late June, and then their chicks fledge in late July. A really important thing to note here is, like many Arctic systems, ice is melting, leaving the system, the ice breakups occurring much earlier in the system. So it's advanced about 17 days, whereas the bird phenology hasn't really advanced quite that much. So there's a lot of opportunity for mismatch in terms of optimal foraging conditions and the timing of the birds and the ice. This system has also seen a really extreme shift in the past several decades from these birds eating primarily Arctic cod, which are an ice-associated species, and again, through the mid-1990s were the primary diet item of these birds, to more capelin. So that is now the primary diet item, which are more subarctic species. And they have similar energy densities, but the capelin are smaller. And so there has been some effects on things like chick growth rates in the system. But even though those are the major diet items, they have a pretty flexible feeding strategy. They will eat benthic and pelagic species and more ephemeral, smaller prey versus larger prey. And so they'll eat sculpin and amphipods and sandland. So there are other things that make up their diet, but the main diet items, again, traditionally Arctic cod, now more capelin. Just for a little more context on this system, there has been actually some previous research looking at diet specialization in this species all the way back in 2008 that found that these birds do show pretty strong individual specialization, and that this specialization is repeatable across time, both in terms of their foraging behavior, so their dive shapes, and then also their diet, as indicated by the isotope values. For an example of this, amphipods only make up about 6% of the diet of the population as a whole, but 76% in this study, 76% of the amphipods were consumed by just 4 individuals. So these individuals are really specializing on amphipods, and you can see that with a lot of the other non-primary diet items in this population. So we know that there is some indication that there's specialization in this population. This study did actually find that there was no difference in the fitness of specialists versus generalists in this population. So that was something that was kind of unexpected and interesting. So we're going to be looking at this same system, but through a slightly different lens today, looking at how murres make optimal foraging decisions and if these foraging decisions basically differ across individuals. So there are 3 predictions. One is that murres spend more time foraging in patches with favorable environmental conditions, right? When there's a higher benefit, it makes more sense to spend more time in that patch. The next is that patches that are further away from the previous patch, there's a higher cost in getting to that patch, so individuals should spend more time foraging there. The next is that individual murres vary in their foraging personality or average or mean behavior and their plasticity. So how flexible their behavior is across environmental contexts. And how we're going to be looking at these data is we're actually going to be aggregating foraging information into hopefully somewhat independent foraging bouts for each bird, and going to be looking at relationship again between some variables of the foraging behavior. So one of the big ones being the time spent in each foraging patch, and then these environmental covariates, and also covariates related to how much time it takes or how much energy it takes to get to that patch. So each sample in this is a diving bout, and then individuals are treated separately, or we'll test for individual differences. So there are 2 things again here we can look at is the behavioral type or average or behavior or intercept of the behavior. And then we can also look at the plasticity or the slope of changes in that behavior across contexts. And the last thing that we predicted was that individual variation would persist across years. So we had 3 types of tools that we use. We use TDRs, GPS, and accelerometers to collect these data. There were 247 murres tagged with 370 deployments over 2017 to 2019. There were 54 individuals that we had 2 years worth of information, and 17 individuals that we had 3 years worth of information. The first thing we had to do was to actually decide how to break up their foraging into distinct units that we could treat as samples. So we decided to use hierarchical clustering with dissimilarity index based on distance. And we wound up going with 1,000 meters after a lot of exploratory analysis using scree plots and things like that. So we use that, and then we use a time cutoff that was developed in previous studies on this system to help separate these bouts. So this is what the output looks like. Each of these colors here is a separate foraging bout. And again, they're separated based on time and space. Once we had these foraging bouts, we collected a suite of foraging parameters and environmental parameters. Just to simplify things, I'm just going to focus on a few today, mainly foraging patch duration, how much time they spend in each foraging patch, and then foraging patch quality. And that is based on a previously developed index that uses things like the amount of time individuals pause between dives and the amount of time they're diving, and it's based around the idea that individuals that exceed their dive capacity are doing so because there's a really good fish down there, or a really good school of fish that they're chasing. And we have found that there is a correlation between the foraging patch quality and the mass of the prey species. So that's kind of been validated in previous studies. And then we used sea surface temperature and chlorophyll A as our environmental parameters and tested whether the days into the season was a better predictor, because this is an ice-driven system where these environmental variables are changing throughout the season pretty strongly. And so we wanted to make sure we were picking up environmental signatures and not time signatures. Once we had all these data, we tested a variety of model structures, so linear versus nonlinear models, and also for the support for random intercepts and random slopes. And once we had the most parsimonious model from that process, we tested for effects of sex and breeding status on the parameter we were interested in. So for example, this is one of the best fit model for the duration in the patch. We have our fixed effects, the environmental parameter, in this case of chlorophyll, the individual intercept or deployment ID, which is personality, and then the individual slopes. The last thing we did was we calculated a repeatability index, which is basically the percentage or proportion of the variation in that parameter due to the individual and not to the other things that, things like sea surface temperature. So at this point, I want to take a moment to thank everyone who braved polar bears and mirror poop and cliffs to collect these data. I have not actually had the opportunity to visit this field site yet, so hoping I get to do so in the future, but a lot of other people were integral in collecting the data that I'm using today. And again, thank my collaborators at Trinity and McGill, and thank the Waterbird Society for this opportunity and for just being such a supportive, wonderful group of people in general.
Thank you so much, Tasha, for your presentation. We do have time for questions. Is there
Sorry, I'm not done. Yeah.
Oh, okay.
I always like to thank people. Do I have some time to continue? I hope so.
Yep.
Okay. I like to thank people in the middle. Sorry about that. Yeah, I'll keep going. So some results now, if you would like that. So the first question was, overall, do mirrors exhibit optimal foraging? And so again, we would predict that they would spend more time in patches with higher chlorophyll concentrations and that were farther away from a previous patch. So we actually did not see that these relationships were supported at the population level. These are the 3 different years. If anything, maybe a little weak negative correlation with distance from previous patch, but definitely not the positive relationships that we were expecting. So I want to dive now more into the individual results. Just check this chat to make sure it's not something about, okay, yes, please, results. Okay, good. So this is the results, including individual ID, and that was supported for this model. So we can see that these individuals vary, jumping ahead of myself, both in their intercepts and in their slopes, how they vary across these environmental conditions. So we do see positive slopes for some of these individuals, but for the most case, we're actually seeing a negative relationship, which doesn't really follow the optimal foraging theory patterns we might predict. There are a lot of potential reasons for this, right? Possibly if they are in a really high quality patch, they actually need to spend less time there because they're able to get food faster. The last thing I'll mention is that, again, of the 3 environmental parameters we tested, chlorophyll A was the best predictor, although the variance described wasn't super high. The other thing I'll mention is that random effects did explain, again, more variance than the fixed effects in this model and in all of the models, really. And that repeatability was actually quite low. So repeatability average across animal populations is about 40%. And in this case, it was only about 14 to 20%, indicating that there is quite a bit of variation within individuals as well. So again, that was duration versus environmental parameter, and we would have expected a positive relationship. Now, if we look at duration versus the distance to the previous bout, we would again expect a positive relationship with the x-axis indicating the cost of making it to that bout. And just like in the previous one, each of these lines is a separate individual. So we see that in this case, random intercepts or individual intercepts were supported, but not random slopes. So all individuals showed a similar slope in this relationship. It described a bit more variance in the duration than did the environmental parameters. Once again, did not follow optimal foraging theory predictions. One possibility here is that when they are sampling, they have to travel farther and might not find a good patch quickly, so we'll spend less time in that patch. But that if they find a good patch, they maybe won't travel quite as far until they find another good patch. Again, no variation in plasticity here, just in the intercepts, and the random effects once again explained more variance than the fixed effects. So far, not seeing what we would expect based on optimal foraging, but there's a lot of good rationale for that based on how this population forages. And the random effects are describing a lot of variance, but there's very low repeatability within an individual. Now, if we look at that index of patch quality that I mentioned, really the main driver here was the breeding stage, so egg or chick. And this goes along with previous research showing that these birds basically are catching larger prey items in the chick stage than they are in the egg stage. And again, prey size is correlated with patch quality in this system. When we looked at patch quality versus the distance to the previous bout, similarly, the biggest driver was the breeding stage, but we did see that individuals varied quite a bit in their intercepts as well. So in this case, environmental parameters were not significant drivers or separate from breeding state, the time into the season, but that breeding state was a really strong driver of the patch quality or predictor of the patch quality. The last thing I'll mention really quickly is that we did not see any correlations in individual slopes across years, but we did see correlations in individual intercepts across years. And this is something that we want to look into a little bit more in terms of looking across behaviors as well and these behavioral syndromes of these species. So take homes, yours spent less time foraging in patches with a higher chlorophyll A and that were farther away from their previous patch, the opposite of predicted based on optimal foraging theory. That patch quality values were largely driven by the breeding state, and that individual variation was more important than environmental drivers, but individuals also varied themselves. So the repeatability was actually pretty low. And the last, that personality was not correlated across years. We saw some evidence that personality or the average behavior was correlated across years, but not plasticity. Next steps, basically just want to point out that these are two very little parts of this big puzzle. And we want to now, look a little bit deeper into these relationships and also start linking them to fitness effects. With that, I'll see if you have any questions.
Yes, thank you, Natasha. We do have time for questions. Okay, well, it's 9:40, so we'll just move on to the next talk. The next up or last up for this session is Kate, who will be talking about dynamics of Black Skimmer migration along the Atlantic coast.
Well, that might happen if I make sure to unmute myself. All right, so can you see my screen pretty well? Except for I should be actually in, there we go, presentation mode.
Yep, perfect. Thank you.
Excellent. So good morning, everyone. Yeah, I just want to thank you for attending my short commentary about spatial temporal variation in black skimmer migrations. This is work that I've been doing for a few years now, and it always seems like every year we add a little bit, piece to the puzzle. So I wanted to first acknowledge my collaborators and my co-authors on this, Tara Schneider and Rob Blanchard from the town of Hempstead. And obviously we had a lot of investment in volunteers and collaborators for this project and continue to. And funding was provided by the Nassau County Soil and Water Conservation District. So it was a really nice project start with collaborations with managers as well as conservation and obviously us researchers. So on the context, we're talking about optimal theories, especially with what Tasha just talked about. My interest in looking at migration is, understanding how migratory behavior and associated routes are optimized. We know that, that this is how most of the studies that we've been doing on have shown that, most of the time the behavior and associated routes are optimized because they want to maximize survival and future reproduction. But there's still a big question out there as to, whether the time or energy, the more influential factor or currency that's used for these birds, and in particularly, and in my focus is the black skimmer. So with that, you think about, if these birds have a time minimization strategy, we're looking at, maybe specific directed behaviors, such as a more direct migration route or faster migration speeds. And when I'm referring to migration speeds, I'm talking about the days on migration. And perhaps maybe increase flight speeds because, time is that strategy. They only have specific time to get somewhere. So they would expect, increased flight speeds during active travel. And then of course, less use of stopover locations. And then the flip side of that is maybe an energy minimization strategy, which we would expect that individuals would have, slower migration speeds, slower flight speeds during active travel, and of course, perhaps increased use and duration at particular stopover locations. But there might be some individuals out there that actually have a combination strategy where they actually are minimizing energy during during autumn migration, where they're minimizing time during the spring. But that requires a lot of information on the how, what, and why dynamics of migration for these particular species we're looking at. So I'm just going to give you a little bit of a background on the skimmer. So the black skimmer is distributed across the Americas, and we've got 6 recognized subspecies. They're colonial ground nesters, and on the Atlantic, for the Atlantic populations, the furthermost breeding population as of this point is in Massachusetts. And then of course they are distributed all the way down to 28 South in Argentina. These guys have a very unique foraging strategy. They're the only ones that employ a skimming strategy where they basically skim across the surface of the water, catching prey items that are within the top 6 inches of the water column. And they have sexual size dimorphism. So the males are about, on average, 100 grams bigger than the females, and that it goes across body size. So, their tarsi, their, the exposed column, their ratios of males are always bigger. And then for the skimmer, particularly in New York, it is proposed. New York has recently proposed to list skimmer as threatened, although we haven't had the final ruling come through on that. But they have conservation status in several other states, such as Florida, where it's a state endangered species. The North Atlantic populations are considered migratory, but they are resident in Florida and in California. And as I found in some research that I was doing in South America, there's considerable variation in migration and dispersal strategies in South America, but we really don't know much about North American movements. So some of the backgrounds on the Atlantic skimmer migration, we know that they basically travel the Atlantic coast, north and south, and they rarely venture inland. For the more northern populations, particularly New York, we know that they depart on their autumn migration, usually September through the first week of October each year. And over the last 4 years that we've been documenting them at the Lido Beach colony, they have left by October 10th to the 13th every year. The spring departure is a little bit more variable. Some individuals start migration in mid-March, and this can extend through early June each year. What we do know through the mark recapture studies is that North Carolina is an important stopover location. There have been individuals that have been noted to stay in November for 3 months, up to 3 months. And we also know from mark recapture studies that Florida is a very important wintering ground for Atlantic populations. So some of the research questions that I've had over there, for this is that, I'm curious about how much variability exists in skimmer migration strategies between autumn and spring migration. Because we know that, going back to that time or energy minimization strategies, that obviously birds have a set physiology and a set ecology, and how can this vary? How do they develop these strategies in order to adjust to these, the endogenous and extrinsic and intrinsic, influences on them. So what I wanted to look at, and there were several things, but mostly I'm focusing on this talk. I'm looking at migration strategies that they're using between autumn and spring. Are they similar? Are the routes similar? Does migration distance differ between sexes? Because there is some sexual size dimorphism occurring with these guys. And is the duration of migration similar between autumn and spring? And then generally getting into some stopover location use. Does the number of stops and the duration of stopover locations vary between the two segments of migration? So my study location, we caught birds at Lido Beach in Long Island, and it is the largest breeding colony in New York. Excuse me. And so we spent 2 years capturing about 46 breeding adults on nests with noose mats. And we have several different projects going on at the same time. So we were collecting samples for genetics, as well as samples for doing some diet characterization. For this particular part of the project, we went ahead and deployed 29 GPS data loggers that were programmed to collect data every 30 minutes from July through August. And then we reprogrammed them prior to migration to collect data every 60 minutes. Although we were actually trying to have them collect less time, but 60 minutes seemed to work for them. And of this, we had 12 males and 17 females that we were tracking. So of the 29 individuals that we had, we had 18 individuals return, with 16 of them providing information for both fall and spring. And then we actually had 3 individuals that provided us with two spring and 2 falls. But for this portion of it, I just went ahead and said, single tracks for each individual. And so what we did was we subset the location data into 24-hour tracks using the move package in R. And then we used net square displacement in package geosphere to calculate a Haversine's distance for movement. Then we went ahead and calculated a route directness factor, which is basically just the total distance divided by the total number of hours on migration to get an idea of whether or not distance route distance was a factor in their migration strategy. And then went ahead and created a linear mixed effect model. And some of the parameters we used obviously were the harvesting distance, or the total distance traveled between breeding location and wintering location, migratory speed in hours. Again, the directness factor, average ground speed per 24-hour segments, number of stopovers, time spent at stopovers. And then we added some more morphometric data like sex, total wing length, mass, as well as, I'm not really going to get into the date departed because I had a small sample size for that. So some of the results that we got were that basically, like as I said before, all individuals departed on the autumn migration by October 10th to 13th, and typically finished migration by early December. So spring migration began mid-March, ended in early June, as we had known from the basic mark recite data. So the interesting thing here, though, is that there was a bit of diversity in strategies that these birds were using. So there were two strategies that we really could identify through the data, and the one is that there's just this direct movement southward to wintering locations, typically in Florida, but also in Cuba. Or there was this nice post-breeding stopover in North Carolina. And it was a very important stopover. Average stopover for the fall for autumn migration was about 41 days for the individuals that chose to stay in North Carolina instead of directly migrating to wintering grounds. And so one thing we did note is that these migration routes are they're pretty narrow. I mean, the individuals use the same route for fall and spring. There's not really any difference between, the route that was used. And it looked like, all individuals moved along the coast until they got to about northern Florida, and then they just basically dispersed. Some individuals wintered along the Gulf Coast of Florida. Some actually wintered, inland in the middle of Florida, and others, a small portion, about 15%, went to Cuba and actually wintered in the part of Cuba that's on the Caribbean seaside. So there wasn't really any sex-related variation in distance traveled that we could find. I mean, the females, autumn versus spring, they didn't have any difference in their movements when looking just at females or males, or even between females and males. They're just, it was very similar in the distances that they traveled. So that was an interesting factor. And then, but what we did see was that there was this incredible difference in the time that they spent on migration. So like I was alluding to earlier, that the average, the mean stopover duration in autumn is about 41 plus or minus 30 days. Spring was only about 27.5. And they had those two direct strategies. We had individuals that actually finished migration fall migration in 28.5 hours. And, or not fall, sorry, that would have been spring migration, 28.5 hours. Whereas fall, it was a little bit, took a little bit more time. So we had some individuals that completed it in like 120 hours, and others that would take up to 90 to 101 days. So it's pretty incredible that there's quite this difference between particularly the time spent actually migrating. And so it seems like that, spring migration was finished in a quarter of the time compared to autumn movements. And another interesting thing that we also noted for stopovers is that 98% of the stopover locations were identified as either beach or shallow submerged vegetation. And they preferentially chose ocean inlets for longer stopover periods. Two of the really important areas in North Carolina we've identified as being Cape Lookout National Seashore, the Core Sound in that area, and then obviously, and then further north up to Cape Hatteras with the Outer Banks. The barrier islands are key points that they seem to enjoy doing stopovers for. And as as we've kind of like I've alluded to, there was no effect of sex on movement, distance, or migration strategies, or migration speed, I should say. So some of my conclusions are is that, you know, we have little pieces of the information and that I think it can support a combo strategy for these guys, that they are, you know, optimizing, you know, they're optimizing energy collection, or I should say they're optimizing. Sorry, guys. They're not worried about time that they spend on their autumn migration strategies, whereas time is a huge component for them for spring migration. And again, North Carolina seems to be a very important autumn staging ground. In the spring staging grounds, we had some individuals that did spend some time in Delaware Bay, but for the most part, you know, it was very minimal use in the spring. And we suspect that the way that these guys are actually moving through the aerosphere is that they're using wind speed and direction to be able to give them that lift northward, so that they're not really moving faster. They're just kind of like using wind support. But that's time for another talk, I think. And the other interesting aspect to this too is that, you know, with such strong migratory connectivity to Florida, it really raises concerns of wintering survival and, you know, and cross seasonal influences upon reproduction, because, you know, we do know that there are some water quality issues that are occurring in Florida. So it's one of the next steps for myself is to kind of continue looking at these wintering movements to, you know, to determine how much impact these water quality and habitat issues in Florida might be affecting breeding populations elsewhere. So the quick and sweet. You guys have any questions? Yeah, I'm available.
Thank you, Kate. We do have some time for questions. Let's see, there is a question in the chat from Jeff. Did you have any indication that birds may have briefly reversed course, doubled back to a more northern staging site during autumn migration?
Yes, we did indeed. We had one individual that was spending a little time in North Carolina, went down to the Charleston, Cape Romaine area. Apparently it was not suitable for him and headed back to North Carolina. For the most part, the routes were generally directed southward. They either stopped and they just kind of like hung out for a while in the area. And that was the only individual that, you know, that we saw this really directed route going north, you know, going back north once it went south.
Thank you. We do have a question from Ricardo.
Hi, Kate. I don't know if I missed this. If I did, I'm sorry. On your movement map, is that for both spring and fall migration?
Yeah, that was just a general map just to show you that how tightly they are. I mean, they basically use the same routes going south as they do going north. Okay. And I was also curious that there's nothing on the southeast coast of Florida. Yeah, it was interesting because they seem to avoid that. They hit Titusville, that area, and then they just kind of like flow inward down into like either Lake Okeechobee in South to Cuba, or they go to the Gulf Coast. Okay, thank you.
There's another question for Kate in the chat from Katie. Have you looked at the effect of age on migration strategy with the tags or mark recapture data?
Hey, Kitty, you know, that is a lovely thing, and I really wish that I could do, and perhaps in the future we can. The part of the issue is that while we've pretty much got almost 100 years worth of mark recapture sites, starting with Gillespie in 1926. It only has been since 2015 that we've really been doing field readables and with any confidence. So we are starting to get information on age, but at this point in time, nobody has been able to, you know, we haven't tracked juveniles and we haven't tracked young individuals because we just haven't had the population, that known age population to be able to test this on.
Thank you. Well, that concludes our session, but there is another session at 10 for those who want to join that session, but we do have this Zoom room available for people who want to ask additional questions or for further discussion with the presenters. But thank you so much to all the presenters in the session today. They were all such wonderful talks. Okay, it looks like people are dropping off to go to the next session, but yeah, thank you so much for participating, and we'll see you for the next session. Have a good remainder of the conference.
Thank you.
Thanks.